All too often, however, the three-dimensional structure itself provides no particular indication of that protein's function. Important clues may come instead from studying how the mystery protein interacts with other known ones. A simple way to identify such interactions involves spotting out samples of a set of known proteins on a microscope slide, and then dousing them with the mystery protein, which has been previously treated so it will fluoresce under UV light. Where our test protein sticks to a particular spot on the slide's protein grid, it has become bound to the protein in that spot, causing it too to become fluorescent. Presumably, then, these two proteins are engineered to interact within the cell. Ideally, to know life's screenplay, to see life's movie, we need to discover all the precise changes in protein composition that occur over the individual's development, from the moment of fertilization all the way through to adulthood. Though many proteins will be found to be active throughout the process, some will prove specific to a particular developmental stage. So in each growth phase, we should expect to see different sets of proteins. Adult and fetal hemoglobins, for example, are subtly different. Similarly, each variety of tissue produces its own profile of proteins. The most reliable way to sort out the various proteins from a given tissue sample is still the long-established method that uses two-dimensional gels to separate protein molecules on the basis of differences in their electrical charge and molecular weight. The several thousand protein spots thus differentiated can then be analyzed with a mass spectrometer, an instrument that can determine each one's amino acid sequence. Unfortunately, to apply proteomics like this to the vast number of proteins coded by an entire genome requires more funding than academic scientists typically have. For the most part, such expensive enterprises are left to the better endowed researchers of large pharmaceutical companies. But because of the method's limitations, even their labs can't routinely find proteins that are present in very small amounts. This type of high-throughput proteomics, with all its expensive hardware and industrial-scale automation of complicated procedures, is therefore not the way most scientists nowadays study gene function at the genome level. Instead, methods of transcriptomics have been adopted because they are cheaper and easier to apply. The functioning of all genes in a genome can be tracked by measuring the relative amounts of their respective messenger RNA, mRNA products. If you are interested in the genes being expressed in, say, a human liver cell, you isolate a sample of mRNAs from liver tissue. This represents a snapshot of the mRNA population in the liver cell. Very active genes, those most heavily transcribed, and that produce many mRNA molecules, will be more abundantly represented, whereas genes that are rarely transcribed will contribute only a few copies to the mRNA sample. The key to transcriptomics is a surprisingly simple invention known as the DNA microarray. Imagine a microscope slide with a grid of 35,000 tiny dot-shaped wells etched onto it. Using precise micropipetting techniques, DNA sequences from just one gene are deposited in each well so that the grid contains every gene in the human genome. Critically, the location on the microscope slide of each gene's DNA is known. Affymetrix, a company near Stanford, has managed to miniaturize these arrays even further by etching them onto a sliver of silicon the size of a small computer chip, yielding a DNA chip. Using standard biochemical techniques, you can tag your liver mRNAs with a chemical marker, so, like the proteins mentioned above, they will fluoresce obligingly under UV light. Then comes the step where the power and simplicity of the technique becomes wonderfully apparent. You simply dump your sample of mRNAs onto the microarray with its minuscule chessboard of 35,000 gene-filled wells. The very same base-pairing bonds that hold together the two strands of the double helix will compel each mRNA molecule to pair off with the gene from which it was derived. The complementarity is precise and foolproof. The mRNA from gene X will bond only to the very spot occupied by gene X on the microarray. The next step is merely to observe which spots have picked up the fluorescent mRNAs. One spot on the microarray may show no fluorescence, implying that there was no complementary mRNA in the sample, and thus we may infer no active transcription of that gene in the liver cell. On the other hand, a number of spots do fluoresce, some with particular intensity. This indicates that many mRNA molecules have bound to it. Conclusion: a very active gene. Thus, with a single simple experimental assay, you have identified every one of the genes active in the liver. And such molecular panoramas have been made possible thanks to the success of the Human Genome Project and the new mindset it has ushered into biology. We no longer need to be content to study bits and pieces. We can now see the whole picture in all its spectacular glory. It is hardly surprising that Stanford's Pat Brown, one of the method's leading practitioners, sees DNA microarrays as a new kind of microscope. Marveling at the technology's potential to reveal a whole new genetic universe, he has declared, "We're toddlers now, just starting to discover our world." Transcriptomics is more than just another brilliant technical innovation. It promises to take us to a new level in the hunt for the genes that cause illness. Using microarray technology, we can discover the chemical basis for particular afflictions by studying the differences between healthy and diseased tissue as a function of gene expression. The logic is simple. We carry out microarray gene expression analysis on both normal and cancerous tissue and spot the difference between the two, the genes being expressed in one and not the other. Once we can identify which genes are malfunctioning, either over or underexpressing themselves in the cancerous tissue, for instance, we may be able to establish a target that can be attacked with pinpoint molecular therapies, as opposed to broadly toxic radio and chemotherapies that destroy healthy as well as diseased cells. And we can apply the same technologies to distinguish among different forms of the same disease. Standard microscopy has offered limited assistance in this task. Cancers that look alike to the pathologist peering through the eyepiece can in fact be critically different at the molecular level. Lymphoma cells, for instance, come in varieties that are hard to tell apart visually, even with the highest powers of resolution. But the differences in their gene expression profiles are clear and vitally important in dividing